that I get the chance here to present some of our space research here at SCK. The first question is maybe why should we do this at SCK, or at least sometimes I get this question, and I thought it's maybe more convincing if this person explains you why than myself. So there is a short video, and I hope it will work to explain what we do and why we do it.
For the moment I'm working here in our European Astronaut Center in Cologne. It's a center where all the astronauts that fly to the International Space Station come and train on the European elements that are flying on board of the International Space Station. And so I'm working here in the center, helping to develop the center, helping to develop the training, but also we are preparing here for the future, because the International Space Station will probably fly till 2020, maybe a little bit later. But after that we need to find new projects, and in space it always takes a long time to build projects, to define projects, and also here at EAC we are participating in that process. Our job is of course very much related to nuclear research, first of all through radiation. Astronauts that fly to the International Space Station have a much higher dose of daily radiation than normal people here on Earth, which means that we need to monitor that, we need to control that also in order to monitor their health. So we have, for example, dosimeters on board of the International Space Station where we measure continuously the radiation environment. But once we will go beyond the ISS to the Moon, to Mars, it will become even more important, because we will not be protected by the Van Allen radiation belt, so radiation will become more important. Moreover, we are also looking into systems that will recycle all the waste, all the human waste, the food for the astronauts. And they will work also with cells, with microbiology. And of course, they will be infected by radiation. And I think the SCK Gen is doing a lot of research in this field. I work together with SCK for some of the research related indeed to what we call the Melissa project. Is this big recycling unit that is based on biological processes, and we flew cells to space. We exposed them to radiation, and then they were examined in SCK to see if their gene expression was changed, if they would alter their behavior in space, because of course if we build something that works perfectly here on the ground, but then we take it to space, go to Mars for 3 years, but after 6 months it doesn't work anymore because all the cells have changed their behavior, then of course it would be a disaster for the crew. SCK is seen as a very important center of expertise in our world. We need to rely, as the European Space Agency, on centers of expertise around Europe. We don't have all the expertise. We can integrate the expertise to build the space station, but we don't necessarily need to have all the expertise ourselves. And in this puzzle, in this mosaic of all centers of expertise around Europe, SCK has a very important role to play.
Okay, so I hope you are convinced that we should do it here. So a short overview of what we do actually is we mainly work to enable manned space flight, and the current most active research is done on 3 domains, which is the radiation dosimetry, the human biology and health aspects, and also the microbiology. The radiation dosimetry is mainly done by our colleague Philippe and our RDC group. They are measuring and characterizing the ionizing radiation inside ISS, but also outside ISS. And more information can definitely can you get from Philippe. But also my colleague Sarah, who is here somewhere, there. They are doing several experiments and projects to investigate the effects of space travel on the human body and the health of the astronauts. But what I would like to present today is the work of our team, which is the microbiology aspects and how we investigate behavior of microbes in space conditions, mainly to assure a healthy environment, but also to develop a system for food, water and oxygen production, and that is what I would like to present or focus further on today. Actually, the main challenges for future human space exploration can be defined as a way to get there and how we should live there. So Further human space exploration means beyond low Earth orbit, beyond ISS, but like what is hype now on internet is these moon villages or a settlement on Mars. But clearly we need still a lot of development on the level of transport and habitats. Supplies, which we call life support, how do we sustain life there? And of course also the impact of space travel on the body, but also on the mind, where you are in a closed environment far from home. What we are focusing on with our group in microbiology is the second part. It's the life support team. And with my presentation, I hope to convince that actually microorganisms are a large part of this big problem. Even if they are small, I'm sure they can help to solve such big problems. Space is in fact a very hostile environment. There is no air, no water, no food. But if we want to go there, we need to breathe, we need to drink, and we need to eat. Astronauts consume daily a number of consumables like oxygen, food, potable water, and also sanitary water for washing, for toilets, and etc. This requires a lot of weight, so about 30 kilograms per day per person. If you then go for long-term missions, this becomes much more. Of course, an astronaut never goes alone. They go in a team, so this is also multiplied then with the number of persons. And it becomes clear that if we want to enable such long-term and further missions, there is a need for what we call advanced regenerative life support systems that are compact installations that actually can produce efficiently oxygen, water, and food in space by sophisticated reuse of the waste. Because if you consume this, of course, you also produce a lot of waste. And again, we believe microorganisms are the solution. And I will present later on the Melissa project to show you how. Currently, the oxygen production and air revitalization in ISS is mainly done through hydrolysis from water with these kind of actually Russian type of machines still. So you provide oxygen typically through a physical technique. In the meantime, all the CO2, which is a very toxic waste product, they are removed on a chemical way by a kind of scrubber cassettes to remove the CO2, so it's a chemical technique. You may remember this from the Apollo 13 movie where they wanted to connect a vertical box to a round hose that was typically to solve actually the CO2 and remove this. But we believe that actually there is a biological alternative for these kind of processes and that we experience every day here on Earth when we breathe. And it's actually the Earth ecosystem, where typically oxygen is produced also from water, but in a process which immediately also removes the CO2, and it's a process called photosynthesis. Photosynthesis is done with the help of light energy, but you need biology. You need cyanobacteria, algae, plants, which actually are using the energy released from photosynthesis processes to fix CO2 to make biomass, edible biomass, so energy-rich biomass that can be eaten. Of course, to make this biomass, you don't only need CO2, but also a lot of other nutrients and minerals, which in principle, in nature, it's coming from the water and the soil and the wastewater. In space, it's actually the key supply is wastewater, and urine is a kind of very expensive, very useful waste stream. And in space, maybe in the future we could also consider to in situ resource utilization or use some minerals from rocks to support this system. So it would allow oxygen, combining oxygen production with CO2 removal, with food production, wastewater purification, and that all with the help of microorganisms. So this concept, this idea is not new. Ever since they started doing flight experiments or flights with humans, this was researched with many different plants, algae, and cyanobacteria. There are examples of systems from all over the world, even from in the 60s in Russia. They already developed the BIOS setup. But also in Germany, France, Belgium, Japan, China, all different systems were developed. There must be said that all these systems were in research and development state. At current, there is no such biological system used in space. Also not the system we are working on, which is the Melissa, which initially was developed by researchers from France and Belgium, but now it's EU-wide. The concept of Melissa is actually based on a lake ecosystem where you have different processes in the water going on, in the deep water where there is little oxygen, little light. There is a kind of fermentation process going on, degrading all the organics. CO2 and nitrogen compounds are released, which are further mineralized. And these kind of nutrients, the nitrate and the CO2, are finally used by photosynthetic systems to grow again. That's how it happens on Earth. The aim was to decipher this and to put it in a kind of system. And actually Max Merget from SCK 25, 30 years ago helped to do this, and we call him one of the forefathers of Melissa. So SCK was founding member of Melissa as well. And a scheme was drawn. The story goes that the very first scheme was drawn on a bierkaartje, a Belgian bierkaartje, but evolved, of course, from that a lot. We have a closed loop system where we use the waste from the crew in a kind of 3 degradation steps or recycling the waste to fertilizers, and then a compartment where we combine plants and autotrophic bacteria to produce again oxygen, water and food. More information you can always find on the ESA website. This project includes many different research aspects and disciplines on the level of ecology, biology, bioreactor technology, because you need to engineer all these separate bioprocesses, technology in gas-liquid separation and loops, in the end you want it to work in space, so you also have to think about conditions like reduced gravity, different gas-liquid separation in these conditions. Luckily, there is a big Melissa team, and you see they work hard to solve all these problems, and when they work hard, they also need to relax hard. But it's a big team to tackle all these problems, and we are a member of it. Since June, we have a Melissa pilot plant in Barcelona where all these bioreactors and all the separate parts we aim to combine. This is one of those typical bioreactors. You see all the tools and supplies, but the real culture chamber is somewhere here behind with this green bacteria. The astronaut in the loop there is replaced by rats that consume the oxygen and produce the CO2, and we have also a plant compartment. Last year was a kind of important year where there was a first closure of the gas loop between this kind of algae compartment and the rats. So the algae provided the rats with oxygen and removed all the CO2 in a hermetically closed system. And the production of oxygen was tuned by also tuning the light and all the regulation and all the modeling and the control. And it worked actually quite well. What will we do in SCK? We work mainly on all the bacteria that are used in the system. We try to analyze how they react and behave in bioreactor setups. We test how they behave in flight conditions and try to help in developing monitoring techniques. And also, on the level of DNA look and also on the level of biochemical composition, look at their potential nutritive value. Of course, the most exciting is doing these spaceflight condition testing. Space has an impact on bacteria, even if they're small. Mainly the reduced gravity causes that the fluid around the cells is actually quite stationary. There is no convection, only diffusion, which makes that waste products and nutrients also are supplied or removed very differently in space, and cells react on that. Of course, there is also the impact of radiation, which can cause damage to DNA, proteins, or membranes, and many other factors like different day-night rhythm or different gravity or gravitaxis in response to gravity. So it's important to know how will the Melissa bacteria react to these conditions, and in the end, how will the bioprocesses be affected. Luckily, we have a number of good space astronauts here on ground who help us to study all these aspects. So we use here in SCK some simulation platforms to test some aspects of space radiation and gravity. We use the facilities of the dosimetry group to combine, for example, different types of radiation, neutrons and gammas. Or if we really want to scream high dose, we use also the BR2 facilities. We use equipment like this, rotating wall vessels or these random positioning machines to simulate some aspects of reduced gravity or this continuously floating aspect. And we combine them, so we put also the machines in the irradiation rooms. But we do also flight experiments, so we prepare these kind of packages here with bacteria on plates or in liquid small chambers. They are launched through ESA, and with ESA help through our Russian colleagues of Roscosmos, so far from Baikonur in Kazakhstan. They typically stay then a number of days in ISS, and where the astronauts are performing our experiments. So you see the packages we sent are then stored somewhere in ISS, and astronaut performs the experiments. After landing, they come back to the lab and we start analysis. And you see we work very seriously and very hard, all together without saying a word, but if you zoom in, at least some people dare to smile a little bit during the work as well. What do we see? We see differences. So bacteria behave different. They show sometimes increased growth rates, some activation of what we call starvation response. They produce some other metabolites. They have a response to altered cell-cell communication. So bacteria talk to each other by producing small chemical molecules, but because they are transported different, also this communication behaves different. Sometimes they change their behavior of biofilms, etc. But the key conclusion is that actually the reaction of your bacteria strongly depends on the growth conditions and the media you use, and also the bacteria. So we need to test this for each bacteria and in the true Melissa conditions. So we are preparing now an Artemis flight experiment together with the European Space Agency and the help of the whole Melissa consortium, which is also financially supported by BELSPO. It's a kind of first bioreactor with cyanobacteria in space to move from small experiments on plate to real bioreactor conditions. And we aim a controlled oxygen and food production in space. This is linked to Melissa compartment 4, which harbors this cyanobacteria Arthrospira. And we try to investigate with this experiment oxygen biomass production in space, and also if the biochemical composition, the food value remains the same. We do this in a photobioreactor setup, which has continuous light, controlled temperature and nutrient dose, and we hope to expose it to real flight conditions. What is Arthrospira? It's actually a blue-green bacteria. So it's cylindric cells which are combined in long strings, which have this spiral form. So that's why it's also called spirulina. On Earth, they like to live at warm temperatures, high pH. They have also these kind of gas vesicles. Yeah, you don't see it here, but which allows them to float or to go down in the water. They can also be motile on a surface. But what is also important is that it's known and used as food supplement already from a long time ago. There are reports that's been used by Aztecs, and it's still used by several tribes in Africa. In Europe, we typically know it as, and also in Japan and China, typically know it as a kind of superfood, and these green pills called spirulina. Why is it so good food source? Because it doesn't contain phycotoxins like some of the other algae. It has a lower DNA content and a thinner cell wall, so typically more easily digestible than some plants or other algae. And it's very rich in proteins, and that's actually the reason why we also want to add it in Melissa, which is a vegetarian system. So we need some good sources of proteins, and spirulina arthrospira is one of them. And it contains a lot of vitamins, minerals, and antioxidants. It has been said that the nutrient value of 1 small milligram of spirulina is similar to 1 kilogram of vegetables and fruits. It depends, I guess, on the way you culture it as well. Nevertheless, several space agencies, NASA and ESA, have adopted spirulina, and they want to add it in the diet of astronauts because of its low weight, low waste, and high nutrient value. Currently, it has been eaten in these kind of cereal bars already by some astronauts in the ISS. Of course, if you want to culture it and you want to be producing high doses of oxygen and produce high level quality biomass, you need to engineer the culture conditions. And we do this via photobioreactors. And we have some of that here in SCK. This is an example of a small one, 2 liters. But for space, even this kind of system you can't send to space. There needs to be another step of engineering. This is what we try to do now within the Artemis flight hardware, a kind of mini photobioreactor, but suitable for space, which we try to build in a 20 centimeter big box with all the necessary engineering of flow, of gas separation. Inside, in the end, there is like a very small culture chamber of 50 ml with a window where we provide light through with LEDs, and we harvest the oxygen in a small gas chamber on top of the culture chamber. This is a lot of engineering coupled to biology, and the big challenge is to do this in sterile way, to work with this kind of electronics, but nevertheless in a sterile way. And you see our group is trained by the people who are making it, Kinetic Q and ESA, to deal with it. If you want, you can always visit the lab to see how it looks, but it's quite impressive. Nevertheless, the people stay optimistic and work hard and try to make it work. And we are preparing for a liftoff in 2017. So I hope with this presentation I tried to convince you that life support will be needed for future flights, and microorganisms can be used, and maybe also on Earth. The last few slides. Okay. So something we discovered along this research is when we tested Arthrospira to see whether it's actually radiation resistant and would survive well in space. We irradiated it with high doses of radiation, gamma radiation in Rita and BR2. We noticed that it actually, we went up to 6,400 grays. Okay, it takes a time to recover, but it nicely recovers even after such high doses of radiation and a full photosynthetic growth. So we are continuing to investigate this, like what is the the biology principle that makes these cells so much more radiation resistant than a human cell or a plant cell. And of course, we are also looking into the potential of using arthrospira as food supplement in radiation protection. Maybe also important to say, arthrospira in our group has led to many activities also to promote communication to youngsters, to attract them to do science, technology, engineering, mathematics. And a big milestone for us was last year with our Food from Spirulina event, where the students, 12 to 16-year-olds, could come to SCK, learn about spirulina, even taste it, and even speak to Samantha Cristoforetti inside the ISS live to ask her what she was thinking about this food bars and if she liked it, yes or no. It was quite exciting experience. And spirulina has been source of inspiration to some other colleagues who developed a project Ondernemers voor Ondernemers to help actually the local spirulina culture in Congo to help to treat malnutrition of children. And they are running a pilot plant, a big bassin with spirulina at BR2. You are always welcome to have a look, and there has been also a call to everyone to stir up the green soup, because it's a culture very basic, totally the opposite of high bioreactor engineering. It's just a bassin, and some people need to mix it by hand. So if you have time, please go by and do it. Some of you already did it very well, and some of them are here. And so this morning, there was the first harvest. They harvested the bassin, and actually it looks very well. You did very well. So there is some green stuff here. Of course, it's the first harvest. We have to check how it looks, what it is. But I heard Maud was planning a tasting. We will have to produce much more to do that, but it looks very promising. And thanks for everyone who already helped, and a call to all to help for the next batch, of course. This is, of course, all work done thanks to the sponsors and the communities we work with, Belspo, Isa, Melissa, the team here in SCK.